Hello and welcome to In Conversation With. This is a series of discussions with people who hold advanced degrees in the life sciences about their careers and career journeys. My name is Chris Hine, and I'm a professor of medicine at the University of Connecticut School of Medicine and the director of postdoctoral affairs at UConn Health. And I'm a former member of the ASBMB Education and Professional Development Committee who developed this podcast. And so we're excited to continue our conversations about the vast array of careers available for grad students and postdocs in the life sciences. Hey, Chandrima.
Hi, Chris. Hello, everyone. My name is Chandrima Majumdar, and I'm a postdoc at the University of California, Berkeley, and I'm a present member of the ASBMB Education Professional Development Committee. We have already recorded several episodes of Careers in the Life Sciences, and they are available on the ASBMB website as well as on YouTube. Back to you, Chris.
Great. So today we're going to continue discussing careers in academia with a particular focus on faculty jobs at primarily undergraduate institutions. And so we're thrilled today to have our guest, Dr. Betsy Martinez Vaz, who is a professor of biology and the chair of biochemistry at Hamline University, which is in St. Paul, Minnesota. Hey, Betsy, welcome. And perhaps you can start by telling us a little bit about Hamline University and your role there.
Thank you, Chris. I am delighted to participate in your podcast today. So, a few cool facts about Hamline. So, Hamline was Minnesota's first institution of higher education. It was established in 1854. Another cool fact about Hamline is that Hamline was also the very first institution in Minnesota to award degrees at the level of bachelor's as well as master's to both men and women. So
Oh,
those are
wow.
two very important things that I always tell people about Hamline.
Mhm.
Other things, we have about 2,500 students, and about 70% of those students are undergrads. And we also have 30% graduate students. Our graduate school is fairly small, and we do our master's degrees and PhDs in some areas, including public administration, education, and business.
Okay.
In the STEM programs, we only award bachelor's degrees.
Okay.
We also are very proud to say that Hamline has a very diverse student body. About 41% of our students are first generation students, and about another 41% of our students identify as people of color. So I like to say that Hamline has a unique blend of a strong liberal arts foundation for undergraduate students, as well as a graduate school. And, we provide a very inclusive and broad education without having to focus only on undergrads or only on graduate students. So it's a very nice institution to work at. So.
Yeah, very nice. So tell us a little bit about your role there now. So you're a faculty member, obviously, that has moved up through the ranks. You're also a chair, but kind of give us a sense of what life is like as a faculty member at Hamlin.
Well, busy. I have been here for 20 years. My teaching load is of about 6 equivalents. So that is 6 courses per year. And
Oh,
typically in biology and biochemistry, it works out to 2 courses with 2 labs every semester. And in addition to that, we are also expected to do service and to do research. So when I look at what my job is, I spend about 70% of my time teaching. And out of that, there is a 15% component of service. I'm the chair of biochemistry program, and I'll talk a little bit about that in a
mhm.
minute. I also, part of my service is doing pre-med advising. So I talk to many pre-meds, and I advise a few clubs on campus, like the pre-med club. And I'm also an advisor for there is a group of Hispanic students, and on and off, I talk to them about, the activities that they have as part of their affinity group. So now, as a chair, what does that entail at an institution like Hamline? So I'm the chair of biochemistry. Biochemistry is a program rather than a department. So because it's an interdisciplinary program, what I do as a chair is slightly different to what someone who is the chair of chemistry or the chair of biology will do. For
Right.
example, all the faculty in biochemistry are either part of the biology department or the chemistry department. So I'm the chair of the program, but I don't have to do faculty evaluations because,
Okay.
those are done by the chair of the larger program. I don't have to
See.
hire adjuncts because that's also something that the chair of the larger program does. So
Okay.
what I do in my role as chair of biochemistry has to do with curriculum design, like what courses should students in this major be taking? Because Hamline is small. So right now I am the only biochemist on campus. Okay. So when we designed the biochemistry program, I was one of the people who said, well, they need to have these particular classes. They need to have this many credits. This is what a biochemistry degree should look like for an undergrad.
Okay.
In addition to that, I am in charge of collecting all the assessment data for the program. So we assess content knowledge, writing, oral communication, and a variety of other things throughout the biochemistry curriculum. So in my job as a chair, I collect that data, and I am also responsible for writing all the program review reports every year. And I advise many of the students in biochemistry. Because it's multidisciplinary, I have colleagues in chemistry who also do advising for biochemistry, and colleagues in biology who also help with advising in biochemistry. But that said, compared to larger programs, biochemistry only have about 30 majors. So it's manageable, which means, yes, I'm slightly busier than I was before I was chair, but I'm not as busy as someone who is the chair of biology, which has like 200 majors, or someone who is the chair of, psychology, which has like, hundreds of majors too. So
Right. And you're still teaching the same amount?
yes, I still teach
Yeah.
the same amount because it's a program, it's not a department. So no, I don't have a course release for doing that.
Okay. So let's talk about your teaching a little bit. So give us a sense of what that looks like. Are you teaching sort of all levels or all grades, all, you have large classes, small classes? What does that kind of look like?
Interesting question. So over the past 20 years, I have taught at all levels. I have taught
Okay.
introductory courses. I have taught first year seminars. Normally, I teach juniors and seniors.
Okay.
So Biochemistry 1, which is one of the courses that I teach, is a course for juniors. Some seniors take it. It normally has anywhere from 35 to 40 students. So
Okay.
it's the course, and then it has 2 laboratory sections. I also teach microbiology, and that's a smaller because it's a course for seniors. It's what we call a 5,000 level class, and it's focused on reading the literature and presenting the literature. So that there I only teach 18 students.
Okay.
Okay. The same is true for molecular biology, which is the other course that I regularly teach. So molecular biology normally has between 14 and 18 students. And it's the same type of course compared to microbiology. Is a 5,000 level course. The students are reading the literature, they're presenting papers. That's the
Uh-huh.
way they're learning the subject. So if the courses are very advanced, like at the 5,000 level, they're normally capped at around 18
Okay.
for both the lecture and the lab. If they are intermediate courses like biochemistry 1, they're normally capped at 40.
Yeah.
And if you are teaching a first year course in chemistry or in biology, like introductory biology or Gen Chem, those are normally capped at around 60
Okay.
students.
No, it's a pretty big class.
Yes. And they will have multiple lab sections.
Mm.
And then the first year seminar is something everyone gets to teach. So those tend to be small because we have students for the first time, right? So those are also 15 to 18 students. So yeah.
I see. And sorry, these are so the more advanced classes obviously are going to be students that are now more focused on the biology, biochemistry majors. But I imagine some of these classes might involve non-majors as well, at least at the beginning.
Yes, and some at the beginning, when you are teaching, even in introductory biochemistry, we have students who are neuroscience majors
Yeah.
or are
Okay.
exercise sciences majors, and they need this introductory or intermediate biochemistry class because they want to go to graduate school or because they are
Yeah.
trying to get into a professional school that requires that particular class. So we also have classes for non-majors, like I also teach a class called Emerging Infectious Diseases, which any major can take at Hamline. So it's a general science requirement. I mean, we have many of them, but the one that I offer is called emerging infectious diseases. People can sign up for it. So, and it's just at a very basic level, just to help with scientific literacy. So
Yeah.
So one of the things you mentioned is that you try to create a learning environment that encourages curiosity for science, its application to daily life, that's kind of like relevant about your biology of infectious diseases class. Can you describe how you go about fostering such an environment for students?
Well, I think I use many clips from the news every day. Like, right now, when we hear about the bird flu, so we talk about, okay, what is this about? What questions do you have about it? And we try to connect it. And another way for the science courses, my students have to keep an eye on the Nobel Prize announcements every October. And
Mhm.
when, like, last year when AlphaFold got the Nobel Prize in chemistry, we had a long conversation about why a program that is computational and has to do with proteins, which are biological molecules, how is that getting a Nobel Prize in chemistry? And we had the same, very similar discussion when the CRISPRs were awarded the Nobel Prize, etc. So, one of my ways is, okay, well, what is happening in the news today? What did you see in social media? What did you hear about on your way to class? And, that's the way for me to connect it to class content. In biochemistry class, right now, we are talking about energy and metabolic pathways. So I talk about diets and what is happening to your body if you are in a high protein diet or in a high carb diet. And, then students have all these questions about, the keto diet and all those things they hear about in their everyday lives. And
Yeah.
another of my favorite things to do is when I'm teaching about biochemistry, I try to compare things to everyday life. Like I have told people, well, if you were at a concert and suddenly you need to get out of the concert, who will get out first? The people that are really small and might get stuck in the crowd, or the people who are tall and big and can push people through? And really, that's my analogy to talk about size exclusion chromatography,
Oh my
right? So I
God, I
tell them,
love
some
that.
proteins are gonna get stuck in during these little parts of the column. But if you are a big protein, you will get to push through. Just like if you were at a concert, you will get to push through because you are just bigger versus someone who is tiny might get stuck. And
Yeah.
and I can tell you many others, right? But I'm trying to
Mm.
find one that, hey, everyone goes to a concert. So you
Well,
can,
it sounds like that must be part of the fun of it, right? Is trying to come
yeah.
up with creative ways to get to your students. And then when you can see the light go on in their eyes, that must be pretty rewarding.
Yeah. So the other day in class, I was also talking about how a reaction with a highly negative delta G has the potential to drive the other reactions on the pathway forward. And many
Mm.
of the kids, their eyes were so big, like, what are you talking about? So I love dominoes. It's like if you have a big domino here and there's a little domino, so you push one, it's a domino effect, right?
Yeah, yeah.
And that was, I mean, for some of them, they were like, oh, okay. So I love doing that. So.
Yeah. So, we're gonna talk a little bit more about your training and your preparation for this later on. But when you're going through grad school and a postdoc, you're very specialized and you may be in the field of biochemistry, but it's usually a very narrow sliver of biochemistry. And now you're doing something where you have to teach, as you described, all these different levels and these different courses. And so I guess maybe comment a little bit on the preparation you have to do for teaching and the ability to stay broad and yet have the specific knowledge that you need as well to narrow in on certain topics.
So I think something I learned very early was to identify what are big ideas in any
Mm,
field.
mm,
Because textbooks are pretty thick, right? And you can very easily get lost in content that is very interesting, but perhaps not instrumental to advancing your knowledge of a discipline, right?
mm,
So every time I have to learn something new, I think, okay, what is the big idea here? And I start out with, perhaps I read the chapter of the book, but then as I'm reading, I'm highlighting what are the big ideas
mm,
that I am going to talk about. If I'm going to teach about glycolysis, what are the big ideas about glycolysis? Rather than, a specific reaction or what are the big ideas. And once I have identified the big ideas, then I try to come up with what would a fun lesson look like? How do I take that big idea into something students can relate to? Is it related to diet? Perhaps can it relate to diabetes and what goes wrong during diabetes? So, to get the student engaged, interested in the theme. Okay.
Yeah.
So other things, if it's something that I have never seen before, or I saw very little of it in grad school, I love using podcasts and TED Talks by people who are experts in the field. That way I can see, number one, what are the big ideas? Because this is a TED Talk or podcast, they are going to be going from the big ideas. They're going to be talking to a broad audience, right? So that really helps me. It's like, okay, these are the points that I need to hit. And then I start thinking, how am I going to hit them? Staying current. I am very intentional about staying current. And Hamline is a small institution, so we have very small departments. But luckily, we are right next to the University of Minnesota, which is a Research One university. And they have all these seminars that you can go to for free. So I have the schedule for biochemistry seminars. I have the schedule for microbiology seminars. Many times, some of these things you can join online because, they put them on the web. So I'm very intentional about that. Like I'm pretty sure I make sure that every semester I'm attending at least a couple of these to see what's going on. I'm also a member of multiple professional societies, including ASBMB. So I get their newsletter. I go to the meetings every year, and that's my way to stay current. I truly do not think you can be an effective instructor if you are not current, right?
Yeah.
And it's very easy to just let go if you work at a small institution and you're just focused on teaching the same thing all the time
Yeah.
until something happens. Like I get emails from my students. Did you hear about the latest protein that is linked to this disease? And did you hear about this other, cancer? I'm like, whoa. So yes, you have to stay current. So
Yeah.
Yeah, kind of riffing off of that point, could you elaborate a little bit about the importance of professional societies and faculty networks that helped you prepare for your present role?
I think I'm glad you asked that question. I think professional societies and faculty networks have been instrumental to my development as a faculty member and also to my research as well. So, for example, I am a member of the American Society for Biochemistry and Molecular Biology, and through them, not only I benefit as a faculty member because they have the education activities at the meetings. They have a very strong education community that, you can talk to all these colleagues and figure out what they're doing in their classes. You can exchange materials for teaching. We even have a conference sponsored by the ASBMB that is focused on education, and it's called Transforming Undergraduate Education in the Molecular Life Sciences. So that is a very strong community. So if you have never taught biochemistry or molecular science before, there are multiple people willing not only to help you, but also to mentor you through your career. They are willing to write a letter for you if you are applying for a job. They are willing to provide you with their teaching materials so you can use them to plan your class. In addition to that, they have all these programs for students. So I am also in the student chapters committee. So my students benefited from that. They got travel awards to go to the national meeting and present the research. They were also inducted in the Honor Society. So, and I also want to emphasize the community. Through the ASBMB, I was able to join a faculty network called the Malay Dehydrogenase QR network. And I have so many colleagues, and we work on similar research projects. And when I need something, if I need somebody to do like a molecular dynamic simulation of the proteins that I'm studying, he will do that. Like he's willing to do that and share the data with me and my students. When I needed mutants that mutant proteins purify, and I didn't have all the resources to purify 10 of them. One of the collaborators in that network said, oh, don't worry, I will send you all these mutants. I'll purify them in my lab. You can have them. And to me, that's invaluable, that I can find not only colleagues willing to share their knowledge and expertise, but also their materials with me. So I think it's fantastic.
That's incredible. Yeah, we spoke quite a bit about teaching and how these networks played into like sharing course materials and helped you plan your courses. Can you talk a little bit about how undergrad research fits into your current job? Like, how do you plan for that? And like, yeah, how do you adapt research plans for undergrad students?
So I think it's a very important part of my job. And we do it at Hamline both as part of summer research and also as part of our courses. So at Hamline, most of our biology and biochemistry courses have a laboratory component that is taught as a course undergraduate research experience, okay, or a QR. So some people might be familiar with QRs. So our students are involved in QRs since their first year. So there's one in introductory biology, then there's one in cell biology, there's one in biochemistry, one in microbiology, and so on. So that's one way that we have to get our students going into research. Now, we also have a very robust summer research program at Hamline, and we support about 60 students every summer. And these are students not only in STEM, these are also students in the humanities and the social sciences. So we have different pockets of money to support about 60 undergraduate students. And we take them to the National Conference on Undergraduate Research. We take them to the ASBMB General Meeting. We take them to other professional meetings. So research is a very relevant part of my job. Now, to your second question, which was how do you create a research program for undergrads, right? Well, I think the first part is to think of the scope of the project you are doing. So when you were a postdoc, you were at a large university. So you will think, okay, so a project will be, we are going to make 10 mutants, we're going to do kinetics in all of them. And after that, we're going to continue doing perhaps some bioinformatics studies or some modeling or molecular dynamics. And initially you think, oh yeah, let's do that project. Well, if you have a small lab with undergrads, you have two options. You hire 2 or 3 undergrads. We have one make the mutants. We have the other one purify the mutants. And perhaps you have the other one work on some of the simulations and dynamics you want to do. But you have to be aware that one student is not going to be able to do all of it. So you have to have projects that have, not as big of as a scope as the projects that you will do in a big research institution. The other part is money, right? Like, how are you going to be able to afford these projects, right? So you have to go with something that most likely will get the students some ownership. So the student feel, I did that is good, even though it might be simple. The project might just be, okay, they made some mutants and they tested for activity and it ends there. But the student did it. So small, affordable. And the other part is think about, can you collaborate with someone at a bigger school or perhaps in a different department? Because that way you can get more money. And then you can, have the student experience different aspects of the research.
So can we talk about this from the perspective of you as the faculty member? How important is research and sort of your evaluation as a faculty member? And I notice you have a number of publications on your CV. How important is publishing to your sort of progress as a faculty member there? Things like grant writing, is that part of the evaluation, or is that sort of more secondary, obviously, to the teaching?
It is certainly an important part of our evaluation, not only for promotion and tenure, but also for advancing to full professor.
Yeah.
However, the expectation is not the same as if we were in a big research institution. The way I look at it at Hamline is you are expected to publish maybe a paper every 5 years, perhaps.
Okay.
So 4 to 5 years, and versus if you are at a bigger institution like the University of Minnesota is a research one, there will be maybe a paper a year or multiple papers per year. Versus here, if you have a paper every 4 or 5 years, I think you are doing really well. And I remember for my tenure at promotion, I needed to have at least one peer review publication or a major grant. If you had both, great, but you needed to have one or the other because it really
Yes.
depends on the field, right? Perhaps if you are in a field in which, in order for you to publish something, you need a lot of money or you need extensive, equipment and materials we don't have. Perhaps you are not there yet, but you wrote a grant and you were able to get funding, so that will
Yeah.
count, right? Now, for advancing to full professor, for that, it's like you have to show, okay, you must have published something somewhere, peer review, and if it involves undergraduate co-authors, even better. So the gold
Right.
standard is not that you have very many, but it's like, do you have any with undergraduate co-authors, right?
Yeah.
And how are you getting your students involved in the research? Sometimes you have only one paper with undergraduate co-authors, but you can show that you took these undergraduates to multiple meetings and they presented the research at multiple conferences. So that also counts as the scholarship requirement. So again, it's a very important part of the job. I haven't seen anyone here being promoted without being published somewhere. So you do
Yeah.
have to be published. So you shouldn't expect that you're going to get tenure or promotion to full
Right.
professor without having peer review papers or without having at least some type of grant. So it is very important. So
Yeah. So then I'm wondering about, you might come, you might be a PhD student or a postdoc at an R1 or a medical school working on a big project, and now you take a faculty job at a place like Hamlin. And I'm wondering if you have some input from your own experience or from other faculty in terms of how you adapt your expertise, which might be in an area that is not really compatible with the smaller university, to be able to be successful, kind of taking advantage of your expertise, but recognizing that you can't do the same things that you might have been doing at your R1 postdoc.
great question. I think there are two ways to go about it. One way is if you work in a system that is very specialized and hard to adapt to the small institution, you can talk about partnering with someone at a larger institution. Like I have colleagues who are partnered with people at the University of Minnesota, or they are
Hmm.
partnered with people at the Mayo Clinic. And basically they do the part that can be done at Hamline, which is normally growing cells under a particular condition, or extracting a protein and having the protein ready to go do, high level NMR on it, or something like that. So that could be a way, because then you become a collaborator for someone at a larger institution, and then you can have the students participate in the project, right? So that
Yeah.
is one. The other one is you need to pick a system that works in the undergraduate institution, right? So if you were someone who work on mice, then think about, well, can the questions I was asking with mice being asked using yeast,
Hmm.
right? That is a simpler model
Hmm.
that can be adapted to the classroom. Or can we use something like zebrafish, which is, we can maintain zebrafish in here. It's not that
Yeah.
hard, right? So you have to be clever about that, especially when you are writing those applications, because the search committee is going to look at that. Like someone is coming here from this fantastic lab at Harvard or at Mayo. Okay, what are they going to do here?
Yeah.
And if they cannot talk about perhaps we have a partnership, or if they cannot talk about, well, I was looking at this, you know, in mice, but I think we can look at a similar system, ask similar questions in yeast, or there is a very similar system in bacteria that has not been studied before. So I will use my
Mhm.
expertise and then translate my expertise into a smaller system simply because it's more affordable. So yeah,
Yeah.
so those are the ways to go about it.
Yep. Very good.
So we talked a little bit about publishing and like how you have to publish a paper once every 4 or 5 years. How does the writing process with undergrads work? Because they're going to graduate in 4 or 5 years, and this is also probably their first experience of learning how to write a research paper. So how do you like handle that?
I think my approach to it, I think it's important to just jump into the pool and try to swim out. So when I'm doing things like a paper with my undergrads, I tell them, okay, here is what it should look like. Go ahead and write a draft. And normally I give them a section. So let's start with a section you know a lot about, like the materials and methods. Go ahead and write that, see what it looks like. And then I ask them, okay, so we have all these figures that we want to use for the paper. for the results and discussion. So I tell them, okay, pick the experiment that you like the most when we were working on this and try to write about what your data means, right? So as I said, I like this approach of like, let's jump into the pool and try to swim out.
Mm-hmm.
This is what the paper should look like. Let's try to write a draft, see how it turns out, right? And then I normally tell them, okay, let's start by doing something that you know very well, the materials and methods, or a figure that you work really hard to get together. Let's do that first. So
Yeah. And you talked about earlier how the research projects are often broken up amongst multiple undergrads that each have their own part. So I imagine the papers are often the same way. It's not like a
yes,
PhD thesis where like you've got the first author who did most of the work and is really writing the whole paper.
no, they do write different pieces of it.
Yeah.
Like, somebody did all the growth experiments, so that person will write about the growth experiments. And then maybe somebody else did a specific activity in some of the mutants, so that person will write about that. And then I'm the big editor at the end. So I start reading it. I was like, okay, let's, move it here and there. And there you go. It takes a lot of time, but
Yeah.
they really appreciate it. So
Yeah,
And we
that's
turn the whole
fantastic.
document red as
Yeah.
the PIs with
yes,
all of our
absolutely.
edits.
Yeah. Yeah. So that said, many of the courses that we teach at Hamlin are writing intensive, and they do write a fair number of lab
Okay,
reports and
that's
a fair
important.
number of mini grant proposals. So, the students normally they have some idea. Oh, it's kind of like a lab report, but it needs to be better. Like, yes.
Yeah.
So
Yeah.
That's awesome. I want to just like shift gears a little bit and talk about the path you took to get to where you are right now. You did your PhD in molecular biology at the University of Minnesota. At what point did you start thinking about a career working at a PUI?
I think toward the ends of my PhD training, I had been a TA like everyone in grad school. You have to TA something. So I was a TA and I enjoyed that. And then I was a postdoc, and I spent a lot of my time as a postdoc teaching grad students and teaching undergrads how to use equipment, how to design experiments. I used to spend all this time like, drawing these diagrams of how things connected together and having these long philosophical conversations about how cool a particular system was and why you should study it. And that's how I knew that perhaps teaching was a really good fit for me. I love telling stories, and I used to entertain the undergrads in the lab with old stories about bacteria and about DNA and the history of these things. And my postdoc advisor one day said, I really think you'd be good for teaching because you spend all this time, taking these undergrads in the lab and telling them stories. And so I think early on as a postdoc, and then I decided that if I wanted to be a faculty member, that then I better start figuring out what to do about it.
Yeah.
And
Mhm.
that's how I found, opportunities to teach. And I did a program preparing future faculty that
Mhm.
was instrumental for my development, not only as a scientist, but as an educator. Like I was in that program before I started teaching. And not
Can you tell us
only
a little
did
more
I
about that?
learn
Can you
how
expand on
to,
that? Can you expand on that program a little bit, maybe?
yes.
Yeah.
So it's called Preparing Future Faculty. Different institutions across the United States have different versions of it. So it might be
Okay.
called something else. Elsewhere, I know the University of Wisconsin in Madison has a program called Scientific Teaching that
Mm,
is for graduate students and postdocs who want to pursue careers in teaching. At the University of Minnesota, it was called Preparing Future Faculty. I learned everything from what a learning objective is, the different ways people learn. You learn how to put together a syllabus for a class. You get a teaching mentor and you go and do a teaching practicum. So you get sent to an
mm,
institution that is like the institution you think you want to work at. And you go there for a semester and you get a mentor and learn everything that faculty members do. Like you go to department meetings, you go to one of the student events, you
mm.
get to teach 3 or 4 classes. And another very valuable aspect of this program was that you learn about the academic job search. So we learn about all the different types of academic jobs, how to apply for them, how to put together a competitive application, how to write a teaching philosophy. So if you are
Yeah.
applying for a job at a PUI, they are going to expect a strong teaching philosophy. You have to teach a class when you go to for your interview. So I remember
Okay.
when I was going for my interviews, it's a 3-day interview, and one day you have to teach a class. So the head of the search committee called me and said, "Oh, we love to have you for an interview, and is on this day, and you have to teach a lecture on photosynthesis." That's all he said.
Mm.
And I said, "Well, what about photosynthesis?" He said, "Whatever you think students should learn about photosynthesis." So it was wide
Yikes.
open. So because I had been to the Preparing Future Faculty program, I knew, okay, this is going to be my lesson plan. These are going to be my learning objectives. This is the active learning activities that I am going to do to show that I can engage the students with the content of the lecture. So I think because of that, because I was very sure that I wanted to teach, I did all I could to prepare myself. And one valuable thing was the Preparing Future Faculty program. Another one was I tried to find mentors. So I kept talking to people who were working at PUIs and asking, "Oh, how did you do this? How did you go about looking for these jobs? Or how did you prepare for the interview, etc." So
Yeah, yeah. Yeah, remind me again, at what point in your training did you do this program?
I was a postdoc.
When you were a postdoc, you did this. So I
I
wanted to
was
ask you, oh, go ahead, go
Mhm.
ahead, go ahead.
I was during the first year of my postdoc when I did this.
The first year of your postdoc.
Mhm.
So I wanted to ask you that program aside, you've sort of answered part of this earlier, but I guess I'm just curious, generally speaking, how important do you think it was to do a postdoc for where you are now?
I think it was very important.
Okay.
It is very competitive to get jobs at PUIs. Many people are applying. And again, it depends on the discipline. Perhaps for some disciplines, those disciplines, it's harder to get people who want to teach at a PUI in a particular discipline. So
Yeah.
perhaps if you are in that discipline, you might not need the postdoc. But I think in the biological sciences or in the
Mhm.
molecular sciences, it's pretty much expected because almost
Okay.
every applicant
Okay.
have been a postdoc. Now, was the postdoc important? Well, it made me a much more competitive applicant that I was a postdoc for a while, for almost 5 years. In
Mhm.
addition to that, I was also, I think, and I feel I'm a much better researcher because of my postdoctoral training. So I was a good researcher as a graduate student, but when I was a postdoc, I got way better. And you build your confidence and you build your expertise as an independent investigator. You spend time doing grant writing, you spend time establishing collaborations with people, and those are things that faculty members do. So if you're coming straight out of grad school, perhaps you help your advisor writing one grant, perhaps you wrote your papers, but I mean, when you are a postdoc, you get to do these things on your own for extended period of time.
Mhm.
And I think to me, that's invaluable. Like, that I was confident, of course I can do this. I was running my own, you know, show with my experiments
Right.
at the bench as a postdoc. There's no reason why I couldn't do it now. So
Yeah. Yeah.
Yeah, you mentioned there were a couple of things that you intentionally did during your postdoc to prepare yourself for this role right now. And so
Mhm.
one of them was the future faculty role, one of them was seeking out mentors. What were some of the other things that you intentionally did to prepare yourself?
So it was very important for me to show that I can work with undergrads and that I had
Mhm.
done projects with undergrads.
Mhm.
So the undergrads that I mentored when I was a postdoc, I make sure that they had something. So they went to a small conference and we had a poster together. So I would
Mhm.
say, oh, even though this was just a small conference in Minnesota, this was the student who worked in my lab. So I could show that I could mentor um, undergrads. In addition to that, I did some teaching on the side. I was not an adjunct, but I was often hired to do seminars for graduate students or do seminars for undergrads. At the time when I was a postdoc, bioinformatics was an emerging field. And I was working in a lab that did transcriptomics and bioinformatics. So a couple times I did these workshops called bioinformatics for biologists or transcriptomics for biology. So I mean, it sounds very generic, but it was for people who didn't know any bioinformatics and how to apply it to these fields. And I will just lead these workshops. And I was doing that to show that I had a genuine interest in teaching. So
Yeah. So your pathway sounds like a pretty good preparation for what you wanted to do. I'm just wondering if there are anything else that you might advise trainees today that are thinking about this, in addition to things you've already talked about.
I think it's very important to get as much teaching experience as you possibly can. Even though I did the workshops, even though I was in the preparing future faculty, the one thing I wish I had had more of was teaching experience. Like I was never an adjunct. So,
Mm.
so if you have the opportunity to be an adjunct to teach a course at any institution, whether it's an online course or a course, that is face-to-face or hybrid, I think that is going to make you a very competitive applicant. In fact, that might be the deal breaker between you and someone who is equally qualified and has not had extensive teaching experience,
Yeah.
right? So you have to know how to teach. And I think about what will you do in your classroom, because these are teaching focused institutions. So
Mhm.
when you come for the interview, they are going to ask you, do you know about active learning? Do you know about team-based learning? Do you know about teaching in general, right? So if you come here and if you come for an interview and your idea of teaching is, oh, I'm going to do a lecture and show people slides, and you don't have any interactive activities, you don't have any group work, you don't have any demos, it's going to be very difficult to get hired, because the people who are into teaching and are very prepared, they can come to the interview, they will demonstrate no one but multiple teaching techniques during the interview lecture. When they are talking to the teaching committee, they should be able to talk about what an inclusive classroom looks like. How do you serve all types of students, right? So if you are interested in teaching, you should learn about that, right? Like, what does an inclusive classroom look like? How do a collaboration with a different discipline looks like? So if I'm hiring you in biology, you're a biochemist, can you collaborate with chemistry, and what type of collaboration do you see happening? Or can you perhaps collaborate with math? What type of collaboration do you think you can bring to our institution? So think of that. Think about,
Yeah.
it's one thing to be passionate about teaching and wanting to be a teacher, but do you know how?
Mhm.
Do you know anything about how people learn? Do you know anything about the latest trends in STEM education, the challenges in STEM education? If you know those things, I can assure you, you are going to be a very competitive applicant. That will set you apart from the other ones. So
Yeah, great.
It's awesome, Betsy. We've covered a lot of ground from your journey to like things that people should think about when putting together their application and the steps you took to prepare yourself around the way as well. So, kind of looking back, if you had a way to meet your younger self, maybe when she was finishing up undergrad or starting grad school, what is one thing you'd like to tell her about how her career would play out?
I will say, work hard for what you love and compete only with yourself, not with other people, because you are unique. And when you are unique and you work hard, your hard work will pay off. So I think put your passion into your work, forget about the noise, and just be perseverant and resilient and patient. Be patient, because good things take time. So I was a postdoc for a long time, and I kept waiting, when am I going to get the faculty position that I want? And I remember when I saw the announcement for the faculty position that I have. So I started reading it, and I'm like, oh my God, that describes me. Like, they were looking for someone who could teach biochemistry and molecular biology, somebody who could use computational techniques and bioinformatics, somebody who could, work on different systems like yeast and bacteria. I'm like, wow, that sounds exactly like what I can do, but it took a while, right? So good things take time. Just keep working hard at them. So
Absolutely. Excellent. Well, Betsy, thank you so much for joining us today. This was really wonderful. Um, just a lot of great information here. So thank you very much. For our listeners, if you want to continue this conversation, you can visit the ASBMB Active site where you can comment on any of our episodes and ask questions and make suggestions for future episodes that you'd like to hear about. So with that, thank you again, Betsy. Thank you, Chandrima. Good to see you all, and take care.
Thank
Thank you
you,
for having
everyone.
me.
Yeah.
Bye. Thank you.